Hello, everyone. And thanks, Pawan, for introduction. I'm Vishnutej Ellur, a PhD candidate from Washington State University. And I'm pretty excited to share a small part of my doctoral research, which is on investigating the roles of defense proteins in plant for sustainable disease resistance. As you all know, that the plant cell wall is always the first blockade for any pathogen attack. In general, it is made up of pectin, cellulose, hemicellulose, and some protein. And pectin is the major component of the plant cell wall. To breach this blockade and also to cause disease, pathogen therefore produce several cell wall degrading enzymes like cellulose degrading, hemicellulose degrading, and pectin degrading enzyme. Polygalacturonase, or Pgs in short, are the crucial pectin degraders and also an important virulence factor in many fungal pathogens. I'll explain a little bit of pectin. Pectin is made up of 4 domains, and homogalacturonone is the most abundant of all 4 pectin domains. It is made up of D-galacturonic acid residues linked by alpha-1,4 glycosidic bonds. This yellow hexagon chain in the middle, you can see this is the homogalacturonone. And it's also called as polygalacturonic acid. So any enzyme that degrades this polygalacturonic acid is called polygalacturonase, or Pg. Pgs, especially the endo-Pgs, are secreted at very early stage of infection. These Pgs cleave homogalacturonone into almost 10 to 15 subunit long chains called as oligogalacturinoids, or OGs, which can elicit various defense pathway. Therefore, pathogen quickly degrades them into smaller and smaller units so that it can't elicit any reaction. To counteract this, plant produces inhibiting proteins called polygalacturonase inhibiting proteins. And in short, we can refer it as Pgip. These Pgip interact with the Pg, and then they inhibit the pectin depolymerizing capacity of Pg. And also they favor the Pg in accumulation of these long OGs, or oligogalacturinoids, because these OGs function as damp, and then they can elicit many defense-related pathways. Why are Pgips so important? Because they are present in all known plant species. Till today, there is no report of any plant species or mutant which lacks Pgip. They have a role in plant defense. There are many, there are many examples where Pgips have been used to confer tolerance to many diseases. Like here, you can see an example where Pgip extracted from tomato stem inhibited Sclerotinia, and there are numerous examples like that. More importantly, Pgips from one species can be heterologously expressed in other crops and still retain the inhibitory capacity. For example, Pgip of common bean expressed in transgenic tomato inhibited Stenocarpella and also Aspergillus. So there are some specific features that makes the Pgip, like these are cell wall proteins with the N-terminal signal peptide, because these are transmembrane proteins. So this signal peptide helps them to cross the membrane and to be anchored to the plant cell wall. They have approximately 10 imperfect leucine-rich repeats made up of an approximate of 24 residue each. And these 10 leucine-rich repeats are organized in 2 beta sheets, beta 1 sheet, which occupies the inner concave, and beta 2, which occupies the outer convex of the protein. Beta 1 sheets are crucial because they are the interaction spot for the pathogen Pgs. I want to use chickpea as a model to study Pgip because chickpea have several health impacts. They're packed with nutrients like dietary fiber, folate, iron, phosphorus, etc. And the chickpeas are not a common allergen and a great gluten-free substitute for many people. They're also rich in plant-based protein. More importantly, chickpeas are environmentally sustainable crop as well, because chickpea, especially here in Washington or Palouse, chickpeas are used in crop rotation along with wheat to improve soil health. Chickpea is a legume and also fixes nitrogen, therefore reducing the use of nitrogenous fertilizer in the current crop or the next crop. They have a moderate to water footprint, chickpea only uses like 500 gallons of water for every 1 pound of chickpea produced, and they have pretty low carbon footprint compared to crops like corn or soybean. They only use or produce like 0.5 pounds of carbon dioxide per pound of chickpea produced. So pesticides and fungicides are the major chemical inputs that can go into chickpea production, and that's where my research plays a small role to try to reduce the fungicide input as well. There is very little information available on chickpea Pgip so far. The only things we know that there are 2 Pgip genes in chickpea, Pgip1 and Pgip2 on chromosome 6. However, my genome search revealed that there are potentially more than 2 Pgips, and the last 3 ones, which I highlighted in red, we do not consider them to be a proper Pgip because they do not possess a signal peptide, and they also lack features that most Pgips should possess. So I propose a new genomic organization of Pgips in chickpea. The old ones, Pgip1 and 2 on chromosome 6, and the new ones, Pgip3 and Pgip4 on chromosome 3. The Pgip2 might not be a true Pgip, and I will let you know why in the coming slides. So here it is a secondary structure of Pgip that shows you the signal peptide and all the loose-enriched repeats present in the protein. Like you can see, Pgip1, Pgip3, and 4 has a distinct signal peptide, and for Pgip2, we don't see any signal peptide. Secondly, all the Pgip except Pgip2 has 10 distinct loose-enriched repeats, which is the crucial feature that makes it a Pgip. But we can see Pgip2 lacks almost 50% of its loose-enriched repeats. Using Swiss model, I constructed a tertiary structure or a model for all the Pgip, and using the most studied Pgip, that is common bean Pgip, as a template. So again, you can see all Pgips except Pgip2 have the concave structure, or the concave distinct structure where the pathogen Pg can interact with Pgip, but Pgip2 lacks that as well. So Pgip2 does not have a signal peptide and does not have most of it loose enriched repeat, and does not have a distinct signature structure to interact with the pathogen protein. So therefore, we don't think Pgip2 might be a true Pgip, and from here on, we focus on Pgip1, 3, and also 4. So with this background, the most important question we ask is, can chickpea Pgips be used to improve resistance against its pathogenic fungi? Let's find out. To assess if there is a differential expression of chickpea Pgips, we used cultivars Dwelly, which is Dwelly is resistant to Ascopita raebi and also Fusarium oxysporum farmer species Caesari, and its counterpart Spanish white, which is susceptible to both Ascopita and Fusarium. We also tested botrytis and Sclerotinia, as there are no reported resistant source or resistant variety for botrytis and Sclerotinia. We considered Dwelly and Spanish white for them as well. So we treated both these cultivars with all 4 different pathogens independently, and at every time point for every pathogen, we had 3 biological replicates. We took leaf samples for Ascopita, botrytis, and Sclerotinia, and we took root samples for Fusarium. And we carefully selected time points for all the different fungus based on literature and also some preliminary analysis we did. So after having samples, we extracted RNA, converted it to cDNA, and we used 2 reference genes, that is 18 srRNA and 25 srRNA. After RT-qPCR, we used the CT value to analyze the results. So the transcript level of each target gene or each Pgip were normalized to those at the 0 time point, and also to those with using the reference genes 18 srNA and 25 srNA. And the fold change was calculated using the 2 minus delta delta CT method. So for every qPCR experiment, it's highly important to choose the right primer sets. And my key guidelines suggest we use only the primers that have efficiency between 90 and 110%. So we were careful in choosing only the primers for all the genes, including the reference gene. They were between 90 and 110%. like you can see for all of them here. So my first experiment was with Ascopita, and for inoculating with chickpeas with Ascopita, we used a mini dome bioassay, which was developed by Dr. Vidong Chen in our lab. The picture on the left, you can see, after spraying the plants with the spore suspension, we covered them with small plastic cups for 48 hours so that they can retain moisture, and then the probability of infection increases. Picture on the right is where you can see dwelly, which is a resistant plant. Even after inoculation, it has very less affected, but Spanish white, which is susceptible, it's totally wilted and it's dead. If you can see the results of how chickpea Pjps responded to Ascopita infection, they responded at low level. The fold change values were less than 5 for all genes. And interestingly, Pgip4, the expression levels in resistant and susceptible remained almost same. Next, we inoculated with Fusarium, and the inoculation technique for Fusarium is a little bit harder compared to Ascopita. First, we grow chickpea seeds in trays filled with perlite. After 3 weeks, we take out those plants, wash them, and then the roots are trimmed to 2.5-inch long. Later, we take these plants and then dip it in freshly prepared Fusarium spore suspension for 15 minutes. And for control, we just dip them in sterilized distilled water. We replant them in a trays filled with 50-50 soil and perlite mixture. You can see these chickpea plants 6 weeks after inoculation, where Spanish white, which is the susceptible one, is totally wilted and dead, and dwelly, which is resistant, is very less affected. The expression levels for Fusarium were better when compared to the infection with Ascopita. All the Pgip is expressed when infected with Fusarium. Very interestingly, and in contrast with the Ascopita, Pgip4 had the highest expression when inoculated or infected with Fusarium, and Pgip1 and 3 also had a better expression levels for Fusarium. And there's a distinct difference. You can see the expression levels in resistance is more than the expression levels in susceptible. The third fungus we chose was botrytis, and for botrytis we grew botrytis in potato dextrose broth for 2 weeks at room temperature, and then after the botrytis is gone, the fungal mat was collected, and then we ground it in a blender, and then using a cheesecloth we filtered it, and then using a hemocytometer we calculated the colony count at 2.5 into 10 power 4 colony forming unit per ml, and every plant was drenched with this suspension. So if you can see, the Pgip1 did not respond at all in both. There's no true resistant cultivar for botrytis. So if you just compare between Spanish white and dwelly, there was no difference in Pgip1 response. Interestingly, chickpea Pgip3 responded pretty well in dwelly, but not Spanish white, and also Pgip4 responded well in dwelly, but not in Spanish white. The final experiment was with Sclerotinia. Sclerotinia inoculation method is quite straightforward, where we take a fungal plug and then stick it to a plant, and then wrap that fungal plug with the parafilm or the plastic tape so that it doesn't fall off. And you can start seeing the disease just after 2 days after doing this. So again, interestingly, you can see that Pgip3 and Pgip4 responded very high to Sclerotinia, but Pgip1 did not. It was similar to botrytis, and Pgip4 had a very high response, and it also crossed 100-fold change mark, and it has the highest response in compared to all Pgips and to all inhibited pathogens. Conclusion, we know that now chickpea possesses more than 2 Pgips in its genome. And we also know that chickpea Pgips are differentially expressed in resistant and susceptible cultivars. And there's a higher expression of all Pgips in resistant host, which tells us it has Pgips have an important role in resistance. And each Pgip responds differently to fungal infection. Therefore, it also suggests maybe that's the reason all crops have more than 1 Pgip. With this, I complete my talk, and thank you for all listening my talk patiently, and I will take some questions if there are any.
Thank you, Vishnutej, for that interesting talk. Now I will forward some questions from chat. Kulveer asked, how do you correlate gene expression, which is a quantitative trait, with the disease reaction, which is a qualitative trait?
How do I correlate gene
Correlate
with
gene
the
expression, which is a quantitative trait, with disease reaction, which is qualitative trait.
thank you. Yeah, that's an interesting question. Basically, our goal is not totally to relate the plant resistance. My experiment goal was to relate specific genes, that is Pgip1, Pgip3, and Pgip4. And we were also trying to understand if these genes are related to resistance at some point. And then our next experiments or next step would be why these are helpful for resistance if they are and then if we can get information, yes, if they are involved in resistance, that's when we can firmly say, yes, these are the ones responsible for resistance. And maybe that's when we can think if it's just one PGIP, then we can call it as a qualitative. And if maybe all of them are involved, maybe we can mention it as quantitative. But it's not the goal of my current research.
Yeah, thank you for answering that. Dr. Seri Flint Garcia asks, how common are the resistant alleles in chickpeas germplasm? Are they common in varieties, land races, or in wild relative?
Oh yes, just like other crops, there are many R genes identified in past, and they are pretty common as well. But it depends on the goal. Chickpea is not a huge crop in US, so right now it's the main focus is on just on Ascopita, but there are not many specific R genes identified here. But for Fusarium and Ascopita, there are several of the genes that are identified, and they're pretty common.
Thank you for answering that as well. I don't see any question in chat, so we thank Vishnutej for this interesting talk and your participation in MU Plant Research Symposium.